Hi, my name is Charles.
And my name is Jeri.
And we decided to do our project on the biotech exhibits at the Tech Museum.
I'm here at the Tech Museum in San Jose, and there are a lot of different science and technology exhibits. So we're going to go check out the biotechnology one.
Because we just couldn't get enough of biotechnology, Jeri and I decided to visit the Tech Museum in San Jose. We learned about some interesting things, including heredity, gene arrays, gene sequencing to find mutations, and gene therapy. We wanted to learn more about biotechnology, but we were also interested in seeing how biotechnological issues are conveyed to the public. The first thing we checked out is a gene sequencer.
So here I am at a gene sequencer, and in this exhibit we're looking at different people who have hemochromatosis, which is a disorder where too much iron builds up in your tissues and it leads to organ failure. And so the way that they're looking for the people who have hemochromatosis is through this gene sequencer, and in this little exercise you're looking for which DNA matches up with the people who are affected and comparing it to those who are unaffected. So I found it. This is where this is where the mutation is.
Wait. In class, we learned how this procedure, known as electrophoresis, can be used to easily compare DNA sequences for identifying parents. It turns out that this is also very important for the identification of diseases, such as hemochromatosis.
So cool.
Next, we stumbled upon this exhibit. Look familiar?
Alright. I'm reading about Ashanti De Silva, who is 4 years old, and she was the first person to receive treatment through gene therapy. And she had severe combined immune deficiency. And so what happened was her white blood cells were taken out of her, corrected, and injected back into her body. And so this exhibit was talking about her experiences and what she thought about it. And she thought that it was a blessing and it was actually a good treatment. And she feels much better afterwards.
One new thing we found interesting was a technology called gene arrays.
So this activity is about gene arrays, and gene arrays are a glass slide that has different genetic makeup, as you can see here, and it is expressed as a series of dots.
We thought this provided a cool new visual perspective on analyzing DNA sequences. It's interesting to see how far we've gone in visually representing something as imperceptible as DNA.
And a cool thing that gene arrays can do is help figure out what the best treatment for certain diseases are based on their genetic makeup. And so we're here at this one activity where you have to determine which of these patients needs chemotherapy. And there is a certain array that will indicate that a cancer will spread. So we're looking at these people's different gene segments of their arrays, and we're going to figure out who it is. So it looks like this pattern matches up. This person will need chemotherapy. This is very interesting.
Finally, we had a hands-on experience at the wet lab. The scientists working here explained how the museum provided ways to engage kids with the biotechnology they learned here. As it turns out, kids receive a card that stores their activities at the tech. Then, they can easily access this information on the internet. This lag segment, where kids are taught how to put jellyfish genes in bacteria and make them glow, is one of these activities they can review at home.
That's awesome.
Doing this experiment helped reinforce the material on genetic engineering that we learned in class.
So in here is the jellyfish gene that glows. It was probably cut and ligated from a jellyfish gene and isolated. And So now we have
The Eppendorf tube she just showed actually contains the plasmid, which has been engineered to contain the fluorescent protein from the jellyfish. Here, you see her mixing the plasmid medium in the bacteria tube using a pipette. Then, she heated the mixture to denature the bacterial cell membrane so that the plasmid can enter.
They're telling us that this process is the same process they use to make insulin and growth hormone.
Then, after waiting for a brief period,
According to the computer, the jellyfish gene is now inside the bacteria.
the bacteria was then spread onto a petri dish containing agarose gel, which provided the nutrients for the bacteria to grow.
So this is what the bacteria looks like after it's been grown. And as you can see under the UV light, the bacteria is glowing.
These 4 exhibits left us with a new perspective on biotechnology and gave insight on how scientists package these issues before presenting their research to the public.
So that wraps up my tech museum experience. I got some brochures about stem cells and genetics and GMOs. So now we're going to head back to the dorm and talk about the applications to Engineering 25. So I hope you enjoyed that little tour of the Tech Museum and the biotech exhibits. Some of the things that we learned again were gene therapy, DNA sequencing, gene arrays, and genetic engineering, and all of it was really exciting to see visually presented to us at the Tech Museum, so I had a lot of fun.
One of the interesting things I think is, in E25 we're kind of bombarded with all the factual aspects of biotechnology, but at the museum I felt that there was much more of a focus on the emotional aspects and kind of the pragmatic kind of implications of all the things that we're learning. At the surface level, putting a transgene into a mouse is no more than just pipetting different liquids and solutes and solvents into different bottles and, maybe centrifuging something and then heating something, but, there's much more going on than just what you see at the surface. But at the same time, I felt that the museum really tried to shelter the kids and the adults that were there from kind of the more brutal reality, and they kind of presented just the nice effects and the pragmatic wonders that kind of we have the luxury to enjoy because of biotechnology, I guess.
So I think it's also really important for children to be exposed to biotechnology at this age through these types of exhibits because it's important for them to see the general impact that biotechnology has on themselves or in the community. And so when they grow up to go to college at Stanford and take an intro to biotech class, they will have that passion that they had as a child and be able to apply that in their problem sets and in tests. So going to the museum was really helpful in regaining that passion for biotechnology that I'm sure all of us have. And so I'm ready for the final now. Bring it on. So I hope you enjoyed this, and thank you for this opportunity.
Thanks a lot.